Good day, everyone. Today, we from Group 6 will be presenting the home-based project module to you. We will start off with the introduction for our home-based project module. This experiment is mainly conducted to be able to identify the different types of microbial growth and also to compare the growth of microbes on different samples. Besides, microbes that are very tiny living things, which can either be unicellular or multicellular, are pathogenic and also useful at the same time. Moreover, microbial growth is actually the increase in overall mass or the number of cells and not the increase in the size of the cell individually. The bacterial growth curve has four distinct phases, which are the lag, log, stationary, and dead phase.
Hi everyone. So the objectives for this home-based project module are: 1. To identify different types of microbial growth, and 2nd, to compare the growth of microbes on different samples of everyday life.
Assalamualaikum. So now I want to explain about the experimental setup for Group A, which is closed system. All plastic containers were ensured clean and dry. Each container was labeled with the name of sample. A slice of bread was put into the container and closed with its lid. Leave it at room temperature for 7 days, and the observation and data recorded was performed every 24 hours. For Group B, which is open system, a scrap paper was labeled with the name of sample. A slice of bread was put on the scrap paper. The sample was left uncovered at room temperature for 7 days, and the observation and data recorded was performed every 24 hours. For both experiment, we repeated with different samples such as milk, rice, pancake, and butter. So now I want to talk about the results. So this is the result for bread with closed system. For day 1 until day 3, no growth of microorganism and no morphological characteristic form, and the texture was spongy and soft. For day 4 until day 7, growth of fungi will form and the colonies increase. Thus, the black spot also increase day per day. Then this is the result for bread with open system. No changes happen except for the physical characteristics. For day 1 until day 3, the texture was spongy and soft. For day 4, the texture was a little hard. For day 5 until day 7, the texture was hard. So we can see from this picture, on day 3, the texture of bread in the container and on the scrap paper was spongy and soft. But on day 7, the black spot almost covered the surface of bread in the container, while the bread on the scrap paper was hard. I think that's all from me. Thank you.
This is the result for milk A with closed system. Based on the results, milk A did not have any changes in terms of groups of microorganisms, morphological and physical characteristics even after 7 days of observation. Meanwhile, the results for milk B with open system is different. There is no groups of microorganisms and morphological characteristics in day 1, but the physical characteristic of the milk changed to jelly-like liquid texture. Starting from day 2 until day 7, groups of lactobacilli were found and the colonies of lactobacilli start to increase until the day 7. For day 2, the physical characteristic of the milk is it got lumpy texture and starts to smell bad. On day 3, clumps of a cheese-like substance also form, yellow spots form, and it smells really bad. For day 4 until day 7, the physical characteristic stays the same, which is the color changed to completely yellow and it produced pungent smells. As you can see in the picture, on day 2, no changes for milk A, whereas milk B started to smell sour and got lumpy texture. For day 4, no changes for milk A, whereas milk B produced pungent smells, color changed to fully yellow and clumps of a cheese-like substance forms, as you can see in the picture.
Next is the result for rice in part A. Rice that was kept in part A is in closed system. Throughout the 7 days, the rice in part A did not have any changes in terms of group of microorganisms and also morphological characteristics. However, the rice in part A had changes happening throughout the 7 days in its physical characteristics. The rice in part A didn't have any changes for the first 2 days, whereas on the third and the fourth day, the color of the rice started becoming yellow gradually and also slimy. The rice is no more white but has completely turned yellow and has become very slimy on the 7th day of the experiment.
Hello, my name is Nur Shahira binti Zulkafli. Next, I will explain about result of sample of rice, which is group B. Okay, on the first day, there are no observation. For second day, rice is a little bit watery. On third day, rice is watery, change in color and has a little mold, which is black spot is found. On fourth day, rice is a little bit hard and has more mold, which is black spot, moldy orange spot, white spot, and filamentous hyphae. On day 5, the color of rice is slowly changed to dark, rice become hard and mold is increased. On day 6, color of rice is darker, rice is more hardened and mold also increased compared to day 5. On day 7, color of rice changed to darkest, rice is more hardened and mold also increased. As we can see here, there are differences between group A and group B on day 3 and day 7. On day 3, the color of the rice in part A has turned slightly yellow, whereas the rice in part B has started to change color and had black spot. On day 7, the color of the rice has completely turned yellow in part A and it is very slimy, whereas more black, orange, and white spot are to be seen on the rice in part B.
This is the result for part A flame cracker in a closed system. As you can see, from 1 day to 7 days, there is no changes in flame cracker part A. Next, this is part B for flame cracker in auto system. As you can see, from 1 to day 2, the set seal was hard, but from the day 3 till 7, the set seal becomes soft, and from 6 to 7, the color slowly to fade out. This is a few picture of flame cracker. During day 3, no changes in sample A, but the set seal for sample B was soft. Next, during day 7, no changes in sample A, but the color remained fade and set seal was soft for sample group B. It can clearly be seen that flame cracker only change in their physical state, but no morphological and no microbial group.
Hi, my name is Nadia binti Osman and I am here to talk about the observation for butter sample. So for the closed system, which is part A, we have noticed that from day 1 to day 7, there is no group of microorganisms and no morphological characteristic found. Based on the physical characteristic, we can see that the butter has started to melt since day 2 until the last day. Next is for the butter sample for the open system. Same as the closed system, there is no group of microorganisms and no morphological characteristic has been found from the first day until the last day. For the physical characteristic, we can see that the butter started to get melt in the day 2. We can see that the pepper started to get soaked by the fat of the butter in the third day. As time goes by, the butter started to be surrounded by ants from the day 5 until day 7. But the most important thing is the surface of the butter has become more bumpy due to the melty state. So this is the butter sample in third day and the 7th day. During day 3, there is no changes in closed system, but the butter in open system started to get shiny due to its melty state. During day 7, no spores detected on both butter, but the butter in open system have been surrounded by ants.
Closed system had different results where the mold is present. This is because the key to mold control is moisture control, where the fungi can survive and grow. This is why the mold cannot grow on bread B because the surface of the bread is dry. Meanwhile, the fungi can survive and grow in the container bread A because the surrounding in the container is moisture. For milk, as shown in the result, milk A with closed system has no changes after observation for 7 days in room temperature. This is because the containers were closed and sealed, which make the milk cannot pick up any flavors and aromas from other foods and no bacteria can get in. Meanwhile, for milk B with open system has drastic changes, where it produces pungent smell, clumps of cheese-like substance and yellowish liquid form on top of it. This is because room temperature is warm and provided bacteria a favorable environment for reproduction and cause them to multiply quickly. The bacteria grow by eating some of the protein and some of the milk sugar and produce lactic acid, which creates casein, increase the pH, and causes the milk to clot and spoil within 24 hours. The lactobacillus population also starts to increase when the amount of lactic acid and pH increase, which make the milk to produce pungent smells and changes in texture and appearance.
Based on the rice sample, we can see that the rice in closed system has no changes in terms of growth of microorganisms. This is because the rice in the container could not receive any particles of the air and surrounding environment to initiate the growth of microbes. But as for the rice in open system, we can see that mold has been increased day by day and reproduced by asexual reproduction. This is because the rice have been left uncovered, so tiny spores in the air will land on the surface of the rice. Therefore, mold will be produced. Some of the example fungi that have been found on the sample is Aspergillus species, Penicillium species, and Rhizopus species. Next, for the plain cracker sample, we can see that both sample in open and closed system shows no groups of microorganisms. This is because crackers are manufactured mainly from flour and water. As for the sample in open system, we can see that the color of the cracker was slowly fade out from bright color to pale color. This is because the cracker was exposed to light from natural or artificial sources. Thus, the exposure of cracker to the lights can result in photodegradation. Moving on to the next sample, which is the butter. Okay, so butter sample in both open and closed system also shows no group of microorganisms spotted. This is because butter has a very high content, means it is not ideal for bacterial growth. One of the reasons butter cannot form any microorganism because butter has been produced with pasteurized milk. Refrigerated butter will not grow bacteria unless it already contains psychotroph. But based on our observation, these bacteria have not been observed since day 1. This is because butter can form bacteria due to a result of contamination.
The conclusion is bread in part A, milk in part B, rice in part B has growth of microbes on it, and morphological characteristic of the microorganism can be seen on the sample. Bread in part B, milk in part A, rice in part A, plain cracker in part A and part B, followed by butter in part A and part B, did not have any growth of microorganism and morphological characteristic related to it after 7th day. However, the sample that did not have any growth of microbes have different in the physical characteristic of the food sample after 7th day, which is the color of the sample and the stature of the sample. The physical favorable condition can be temperature, pH, and osmotic pressure, whereas the chemical favorable condition might be carbon, nitrogen, hydrogen, sulfur, phosphorus, and oxygen. The growth of microbes also on different samples were compared to be able to identify the morphological characteristics that were present on the food sample at the end of the experiment. So, that's all from us. Thank you.